So in this video, I'm going to discuss about eukaryotic and prokaryotic DNA replication. So the first process here is the initiation. Okay, initiation. So DNA replication begins at a specific sites called the origin of replication. Eukaryotic genomes have multiple origin distributed across their chromosome. Proteins like origin recognition complex (ORC) bind to this origin and mark them for replication initiation. The second step is the unwinding. Once the origins are recognized, helicase enzymes are recruited to unwind the double helix structure of the DNA at the replication fork, creating two single-stranded template for replication. So this is the helicase. You can see here it unwinds the double helix structure of the DNA at the replication fork. Next step is the primer synthesis. Primase enzyme synthesizes the RNA primer. Primase enzymes synthesize RNA primer complementary to the single strand DNA template. These primers provide a starting point for DNA polymerase to initiate DNA synthesis. DNA polymerase extend the primers by adding complementary nucleotides to the template strands in 5' to 3' direction. Eukaryotes have multiple DNA polymerases involved in different aspects of replication, including replicative polymerase, example polymerase alpha, delta, and other repair polymerase. Then the next step is the leading and lagging strand synthesis. The DNA replication is semi-conservative, we already know, meaning each parental strand serves as a template for synthesis of a new complementary strand. As the DNA helix unwinds, one new strand called the leading strand is synthesized continuously in the 5' to 3' direction towards the replication fork. The other strand, that is the lagging strand, is synthesized discontinuously in short fragments called the Okazaki fragment. So the next step is Okazaki fragment processing. RNA primer in the Okazaki fragment are replaced with DNA by DNA polymerase delta DNA pol. DNA ligase then join the adjacent fragments to create a continuous strand. Then the finally next step termination. DNA replication terminates when the replication fork from neighboring origin meet and fuse or when they encounter a specific termination sequence on the DNA. The newly synthesized DNA is packaged into the chromatin with the help of the histone proteins and the other chromatin remodeling factor. Okay, next prokaryotic DNA replication. Here also the processes are mostly the same as eukaryote, like in initiation, elongation, then termination. But the replication begins at a single origin of replication, single ori in prokaryote, and for that reason is called ORIC region. This region contains specific sequences recognized by the initiator protein such as DNA-A, which bind and unwind the DNA to initiate replication. This helps in initiating replication, prokaryotic replication. This is the specific properties of a prokaryotic DNA replication. Helicase enzymes are recruited to unwind the double helix at the ORIC region, creating two replication fork. Then primase enzyme synthesize short RNA primer complementary to single strand DNA template, just like eukaryote. Okay. Then DNA synthesis, DNA polymerase 3. Here, one thing is that DNA polymerase 3 do most of the work. Okay, so so this DNA polymerase is synthesize new DNA strand by adding complementary nucleotide to the template strand in 5' to 3' direction. Unlike eukaryote, prokaryote typically have only one primary replicative polymerase, that is DNA polymerase 3, responsible for most of the DNA synthesis. Okay, DNA polymerase 3. Then, similar to the eukaryote, one strand, the leading strand, is synthesized continuously in 5' to 3' direction towards the replication fork. Then the other strand, the lagging strand, is synthesized discontinuously in short Okazaki fragment. Then RNA primers in the Okazaki fragment are replaced with DNA by DNA polymerase. Then the DNA ligase then join the adjacent fragment to create continuous strand, just like in eukaryotic DNA replication. Finally, replication terminates when the replication forks from the opposite direction meet at a specific termination sites on DNA molecule. In prokaryote, newly synthesized DNA is rapidly condensed and organized into a nucleoid region, which lacks the membrane-bound nucleus. Here are some basic similarities or differences between prokaryotic and eukaryotic. You can see here recognition of origin of replication is done by DNA protein in prokaryotes, but in eukaryotes it is still not clear. The unwinding of DNA double helix is helicase in both the cells. Then the stabilization of unwound template strand is also single stranded DNA binding protein. It's same. Then the leading strand synthesis DNA polymerase 3, leading and lagging strand is synthesis done by polymerase 3 in prokaryotes, but there are different polymerases for different functions in eukaryotes. Then the replacement of RNA with DNA in prokaryote DNA polymerase 1, but in eukaryote it's still not clear. And you can see DNA ligase is same function in both the cells. And here you will find the schematic of the basic prokaryotic and eukaryotic DNA replication. So that's all in this video. Thank you for watching my video. In the next video I will discuss the other topics of molecular biology.